What molecular mechanisms create immune memory? Imagine your immune system as a smart security team that not only fights intruders but remembers them for next time. That's what immune memory is all about. When your body encounters a pathogen, like a virus or bacteria, it doesn't just fight it once and forget. Instead, it learns from the experience to respond faster and stronger if the same threat shows up again. But how does this happen at the molecular level? Let's explore. First, when a pathogen enters your body, special cells called antigen-presenting cells, or APCs, spot it. These cells engulf the pathogen and display tiny pieces of it, called antigens, on their surfaces. This acts like a badge, showing other immune cells what to look for. Naive T cells, which are like fresh recruits that have never seen this badge before, get activated when they recognize these antigens. Once activated, they turn into effector T cells that attack the infection. Some of these effector T cells don't die after the infection clears. Instead, they become long-lasting memory T cells that stay in your body, ready to jump into action if the same pathogen tries to invade again. Meanwhile, B cells, another type of immune cell, are stimulated by the antigens to produce specific antibodies. These antibodies are like targeted missiles that neutralize the pathogen. Some B cells become memory B cells, which remains in your body for years. If the pathogen reappears, these memory B cells can rapidly produce large amounts of antibodies, stopping the infection before it even starts. Now, at the molecular level, a lot is happening behind the scenes. Key transcription factors, like October-1 and OCAB, help keep immune response genes in a poised state. This means the genes are ready to be turned on quickly when needed. This readiness is maintained through epigenetic modifications, which are chemical changes to DNA and histone proteins. These modifications make the DNA more accessible, allowing transcription factors to bind easily and activate the genes needed for immune responses. Supporting these changes are shifts in cell metabolism. When immune cells are activated, they switch to a faster energy mode to produce signaling molecules called cytokines and chemokines. Some of these metabolites influence enzymes that modify chromatin, linking energy use to gene regulation. This helps keep immune genes ready for rapid activation. During an immune response, T cells multiply rapidly in a process called clonal expansion. After the infection is gone, most of these cells die off to prevent too much inflammation. But a select few survive as memory cells. These cells can live for many years, sometimes a lifetime, and can respond quickly if the same pathogen tries to attack again. In summary, immune memory is created through a series of steps. Naive T and B cells are activated by antigen presentation. They differentiate into effector and memory cells. Epigenetic modifications keep immune genes ready for quick activation. Transcription factors maintain this poising, and metabolic shifts support the process. The surviving memory cells are like a built-in alert system, ready to protect you from future infections. This molecular setup is what makes vaccines effective, and why your body can remember and fight off diseases more efficiently over time.